It's now my pleasure to introduce somebody who I have referred patients to but have never actually met in real life before. Dr. Paul Froomes is a specialist physician, board certified in gastroenterology and endoscopy, completed a postdoc degree in liver research from Melbourne University, has an honorary appointment at the Austin Hospital and is currently the director of the Melbourne Faecal Transplant Clinic, very groovy name, the Centre for IBS Solutions, and is head of the newly formed National Gut Biome Foundation. He's published research papers in peer-reviewed medical journals, presented research at national and international conferences, and the full disclosure is that I've referred people. I am very fond of the idea of fecal transplants these days, and I think they go way beyond what anyone's going to claim. I think there's a future that we are yet to really discover about that part of the gut. So welcome to Dr. Paul Froomes, Fecal Transplants, State of the Art in 2017.
Nice to meet you too. Thanks very much. Good morning, everyone. Sorry, I feel a bit like a principal standing behind that, so I'm just going to come out here. And thank you very much to Thomas for that talk. It's really refreshing to hear a doctor talking about the gut barrier. So I guess it was nearly 10 years ago, when I was working at a public hospital in Melbourne, and we had a middle-aged man dying in intensive care. He had pseudomembranous colitis, which we all know is caused by Clostridium difficile infection. So he had developed circulatory failure. He was being kept alive on inotropes and fed through a nasojejunal tube. Fecal transplant at that time was on nobody's radar. There were actually multiple publications showing that it was effective in C. diff and pseudomembranous colitis, but just wasn't thought about. It took one of the registrars to sort of put his hand up and say, do you think we should try fecal transplant? And look, this was a last-ditch effort. This guy was dying. All the units very hastily agreed, so we've got nothing to lose. We got permission from the family, got his wife in, got a sample from her, and literally did what we do now. We blended it up, made a transplant, and instilled it down the nasojejunal feeding tube in this dying patient. Now, within 24 hours, the bloody diarrhoea, which was continuous, had completely stopped. Now, within 48 hours, weaned off inotropes. Within 3 days, this patient was back in the ward. Now, I've never seen anything like that. None of the heads of unit really believed it. It was a shock. Now, that got me interested in fecal transplant. I thought if it can do that, then the sky's the limit. So here we are on a Sunday morning talking about the magical healing powers of poo, right? Right, okay, well, what's a fecal transplant doing? Well, obviously, we believe it's changing the makeup of your microbiome. We now accept, and this is a quick refresher for all of you, that the microbiome is a vast organ. Well, not an organ. Medical terms, we call it a tissue. Not quite an organ, it hasn't got that status yet, but it's huge. It weighs 2 to 3 kilos, as much as your brain. It's as metabolically active as your liver, which is your powerhouse of the body. Consumes 20 or 30 grams of carbohydrate, 10 of protein a day. This is a functioning organism, tissue of organisms. We call the cells, the microbiota, they outnumber our cells, as you know, 10 to 1, and the genes in the gut bacterial genome outnumber our own human genes 100 to 1, and these are not redundant genes. These are actually coding for proteins. Now, we're not going to touch on the virome, but you do have a gut bacterial virome. It's estimated to be in the volumes of quadrillion viruses, so we'll see something more about this in the future, but we're going to confine our talk to the microbiota today and manipulating it. Now, if you're healthy and you've got a great functioning gut, you live in this space. This space is probiosis, and this is where your gut microbiome is made up of probiotic species, right, that are functioning well and keeping you healthy. Now, it's nearly 30 years since that hygiene hypothesis was first stated, which states that a loss of contact with your probiotic gut bacteria has a direct causal relationship to this epidemic of allergies and autoimmune diseases we're seeing now in the West. And the old friends hypothesis explains that, well, since we crawled out of the primordial ooze as Homo habilis over 2.5 million years ago, we have evolved or co-evolved with our microbes to form a beautifully functioning, beautifully adapted symbiotic organism. We need them. We don't function properly without our gut microbiome, and they need us. So they don't just make goodies for us. Your gut microbiome, your bacteria, interact with your body at an epigenetic level. That's how close the relationships become. We all know they make nutrients, vitamins, minerals, short-chain fatty acids, amino acids, mucins, etc. Look after us. They modify body composition, even behaviour and mood now. But the most important role, we think, is probably that effect they're having on our gastrointestinal-associated lymphoid tissue. So they moderate your gut immune system, as we've heard a bit about, to teach you to tolerate what you're eating and which bacterial antigens that are probiotic in your gut. Otherwise, you live in this space. This is dysbiosis, which you all know about, when your microbes, the composition of your microbiome is made up of dysbiotic bacteria, not probiotics. In this situation, it is very common in the West because it's driven by a diet that is low in fibre, a diet that is high in sugars, and a diet that's high in xenobiotics, those 3,000 different chemicals that are added to food that range from preservatives, emulsifiers, right through to antibiotics that we pour down our throat. These chemicals wipe out your probiotics and you get overgrowth of bacteria that thrive in that environment. Now, you lose all the benefits of your probiotic microbiome because dysbiotics make toxins, right? We know there's a lot of them. There's over 150 of them described: amines, aldehydes, hydrogen sulfide, lactates, lipopolysaccharide, endotoxin. It just goes on and on. Now, these substances are noxious and they disrupt and destroy your gut barrier. And that, hence, you get destruction of the tight junctions, abnormal permeability. And this is a situation that is referred to as leaky gut. But as you know, doctors don't use that term. It's abnormal permeability, right? This is what happens when you have abnormal permeability. You've damaged your mucus, you've damaged your tight junctions, you get translocation. And translocation just means that you get movement of all these bacterial antigens, endotoxins, fermentation products, noxious substances. They can get in contact with the gut epithelium directly and even penetrate it, get to the submucosa, lymph, bloodstream, the whole lot. Now that, you know about the gut immune system, you've heard about it. Our first defence, sitting right under the epithelium, is your innate immune system. This is our first responders. These are antigen-presenting cells, constantly scanning the epithelium and what's coming in. Is it nice probiotic antigens? Is there any damage to our gut lining? No? Great. They will then put out on the MHC platter to the T cells that are scanning all the antigen-presenting cells. Okay, that's a good bacteria, there's no damage, no co-stimulatory molecule, tolerance. We have a nice, healthy, tolerant immune system in the gut, and we're enjoying our environment, we're enjoying our probiotic species, harmony. But what happens in this situation is that we get dysbiotic bacteria. So we get nasty bacterial antigens on that MHC platter. We get evidence of damage to the gut lining, so there's bits of Golgi, bits of endoplasmic reticulum, damage. And then a co-stimulatory molecule that says to your T cells, let's fire up. And your T cells, as you know, differentiate from Tregs, when everything's calm, into Th1, Th2, 17, and the whole inflammatory cascade then proceeds. Now, that's okay if it's a momentary gastroenteritis. That's our defence, right? We're going to attack, get over it, and return to a state of immune tolerance again. But if you have chronic gut bacterial dysbiosis, chronic abnormalities of your permeability, this abnormal inflammatory process, immune process, is going on all the time. This is when it becomes a problem. It spreads right throughout the gastrointestinal-associated lymphoid tissue, up to the liver where the Kupffer cells get activated, and then it can spread to other organs. And that's thought to underlie perhaps allergies, hay fevers, autoimmune diseases. This is where we're heading with this. Okay, well, what about if we could change dysbiosis and improve gut permeability? Might that cure some of these diseases that affect modern man? So how do we do that? There's not that many ways we've worked out yet. We can change the diet. You can eat a prebiotic-rich diet, which will, of course, feed and nurture the growth of probiotic bacteria because they like prebiotic foods, vegetables, non-digestible fibres, right? We can take probiotic capsules. There's a few strains you can get, but as there's thousands of different species in the gut, we can only get a few in probiotic capsule form and a few from eating probiotic foods. Problem is, this study shows that as soon as you stop eating prebiotic foods, stop taking probiotics, they disappear from your gut microbiota within about 8 to 10 days. So that can't change the fundamental composition of your resident microbiome, but they can provide a wonderful day workforce to your microbiome, which has health benefits. So maybe, well, let's look at fecal transplant. Why? Well, because in theory, you're taking the resident microbiota from a healthy person and infusing that into somebody who has dysbiosis. Maybe that'll work better. We didn't invent FMT. It's been going on for a long time. It was first written up in 4th century China by a desperate Chinese physician who was trying to deal with people and kids dying of dysentery. And he used a fecal suspension that was taken orally called yellow soup to cure them. It was rewritten up again in 16th century China, same thing. Vets have been using it since the 17th century. They call it transformation to cure animals with diarrhea. The Germans were described as using camel feces suspension to treat dysentery. And of course, the first report of fecal transplant to cure pseudomembranous colitis will hit the medical literature way back in 1958, and it's been pretty much ignored since then, apart from in the last decade when C. diff has become such a global problem. So what is an FMT in 2017? Well, it's still pretty much what it was when it was published back in 1968. We get a fresh stool specimen, we mix that with saline, put it in a standard blender, gently blend it, then filter out the particulate matter, and you're left with this fecal supernatant, which contains all the goodies. Before we get to that phase, and I'm sorry that hasn't come out too well, but I'll explain it quickly. Before we do a fecal transplant, we have to screen the donors, right? And that was published, the Amsterdam Protocol was published in 2013. There's been a few additions to that since. But the left-hand column, which you can't read, is a detailed medical questionnaire and examination. Put simply, your donors have to be completely healthy. They can't have anything wrong with them, mentally, physically, body weight, nothing. If there's a tick against any of those, they can't be a donor. They're super healthy people. Then if they pass that, they go on and do all the blood testing to look for any potential infective agent. If they pass that, they go on and do the stool testing for viruses, pathogenic bacteria and parasites. And if they pass all of that and they agree to lead a healthy lifestyle, then they can become a donor. So we get 20 donors for every 100 people we screen. And they are bloody hard to find. So what's doing the heavy lifting in our fecal transplant? We still think it's the microbiome. We still think it's the bacteria. Where are we at with that? Well, 98% of your microbiome is made up of just 4 phylotypes. It's predominantly Firmicutes with some Bacteroides, a few Proteobacteria and Actinobacter, and then 2% is made up of this collection of Fusobacteria, a new species called Archaea, and Verruco microbia. So we're not going to break that down to genus or species. We'll be here all night. But that's what's in it. And that's what we think is doing the magic. But be aware that there is a virome, which you'll hear more about in the future. The most well-studied viruses are called the bacteriophages. We've known about them for 30 years. They've been used in the food industry for 20 years. Why? Bacteriophages selectively kill individual pathogenic bacteria. So you can get a bacteriophage that kills enterotoxigenic E. coli, Salmonella, Strep, Staph, Shigella, so that you don't get contamination of food. Well, they're also in the gut, helping you. That's part of your defence. This study was done years ago. They gave mice a gavage of enterotoxigenic E. coli with a probiotic, Bifidobacterium longum. And they all got diarrhea. It lasted for a week or so. If you then added a bacteriophage that specifically kills E. coli, within 24 hours the diarrhea stops. And you get a 100-fold increase in the probiotic levels and a 100-fold decrease in the enterotoxigenic E. coli. So these guys are pretty powerful, and we'll probably hear a bit more about that. But they're targeted. They're not like antibiotics. They don't have a mass effect. There's also some antimicrobial proteins, which we heard about in the previous talk from Thomas, that are secreted by specific Paneth cells in the epithelium and by some of the white cells. Small cysteine-rich peptides that will bind to a cell membrane protein and disrupt the cell membrane and kill bacteria. It's part of our defence against dysbiosis. They don't have a mass effect either. Very targeted. There's also some nutrients. So short-chain fatty acids we all know about, produced by our probiotic bacteria fermenting non-digestible fibres and starch. And they're incredibly important little fatty acids. So they provide an energy source for your epithelial cell function, so they increase gluconeogenesis, right, in your epithelial cells, equals more ATP, better cellular function in the gut. But they also have an effect of inducing tight junction proteins to improve your permeability defects. They induce satiety, improve insulin sensitivity in the liver. They get there through the portal vein. and they help send a nice, calm, stimulatory signal to the gut immune system to tolerate. Treg cells, tolerance in the gut immune system. So you're happy with your bacteria in your food. And the final thing we've discovered in the fecal transplants and the gut is these little microRNA particles. They're about 18 nucleotides long. And these little guys block messenger RNA. So the messenger RNA can't get the ribosomes to construct new proteins. And they're very, very specific. So the most commonly studied one is MIR223. This one blocks protein synthesis of one of the 3 cytokines that makes up the gut inflammasome. So your innate immune system pumps out these cytokines, and there's a trimer of these 3 that are highly potent and drive inflammation in the gut. So it blocks that. You can't make that. And if you give it to colitic mice, the colitis disappears. We haven't seen that used in humans yet, and they also stimulate probiotic growth. Okay, so we still believe that the effect of FMT is basically to restore the richness of your microbiome, so the biodiversity, and the mass of your microbiome. And therefore, that's going to improve the function of your gut microbiome. And in the case of C. diff, it's going to cause colonization resistance. It's going to stop them colonizing your gut. And it reset your immune system to tolerance. But there's likely a bit more going on than just the bacteria. Does it engraft? Does a fecal transplant engraft? Well, the 16S RNA study showed that the recipient microbiome resembles that of the donor at about 2 weeks. And it lasts 6 months. That's in this study in Clostridium difficile. And it does restore the structure and function of the microbiome, and it gets rid of C. diff because antibiotics fail. Right? These researchers at University of Alabama looked at us at 2 years and found the microbiome still resembled that of the donor at 2 years. And the most resilient of the species from the donor that seemed to take hold for that long period of time are the Bacteroides species. But every person who has a fecal transplant has a very individual microbial pattern. So you all know that we all have our own individual microbiomes. They're as unique as your fingerprints or your retina. Even after fecal transplant, it's not the donor's microbiome you've got. It's a composite between what's left of yours and what's taken from the donors. And it's still unique to you. Okay, so we got interested in fecal transplant because of our overprescription of antibiotics, right, which you all know is a problem. So they destroy the mass and the biodiversity, antibiotics, so they'll knock off the Firmicutes and the Bacteroides predominantly. Now that's a problem. You don't have any resistance to infection and parasites, bacteria and pathogens anymore. You're no longer protected by your microbiome. The broader the spectrum the antibiotic, the more damage to your underlying microbiome. And we've known for a long time that up to almost 40% of patients can get antibiotic-associated diarrhea. Now, this is not C. diff. This is C. diff negative antibiotic-associated diarrhea. We know this is dysbiosis that's driving it. This can happen up to 3 weeks after you've finished an antibiotic. Now, if you're lucky, your old microbiome will grow back and repair within a few weeks. Or it might take a few months. Or it may never, ever be the same again. You may wind up, you've all seen this, post-antibiotic chronic IBS. And that's dysbiosis, right? So C. diff is one of the biggest problems we face with broad-spectrum antibiotics, right? So it's escalating. 3 million cases a year worldwide, and that's continuing to grow. If you stay in hospital more than a week, you increase your risk of C. diff by 5-fold. Once you've had one infection with C. diff, you've got a 20% relapse rate. This is after treatment and eradication. If you have 2 infections, that doubles to 40%, 3 infections, 60% relapse rate. And the problem is we're not reconstituting the microbiome after we've treated you with oral vancomycin or fidaxomicin or flagyl. And so this is 4 years old, this data from the States, 250,000 cases, 14,000 deaths. So it's becoming a problem. Where are we at in 2017 with C. diff? It is now accepted as a standard of care in this country, fecal transplant, for the treatment of C. diff. This study is 4 years old now, but people aren't studying it anymore. 325 cases of resistant recurrent C. diff treated with fecal transplant. The mean worldwide cure rate's 91%, which is just staggering. There's not a crusty old professor of gastroenterology that can argue with this data. And by God, they tried. Anyway, it seems to work a bit better if it's a colonic infusion rather than a mid-gut infusion. And it seems to work a couple of % better maybe if it's a related donor than an unrelated donor. So now you would think, obviously we're turning our attention to inflammatory bowel disease given the spectacular results we see with C. diff. Because we know that inflammatory bowel disease is an autoimmune disease. It's dysregulation of your gut immune system to the resident dysbiotic gut bacteria. So there's a 3-hit hypothesis currently to explain the onset of autoimmune disease. Hit 1, a genetic predisposition, and that's very well described for all autoimmune diseases, which includes your HLA and your SNPs. Hit 2, we believe, is a change in the makeup of your gut bacteria. So dysbiotic gut bacteria, we think, might drive autoimmune disease. And it's been described quite well in IBD. It'll be described much better when we get better techniques for analysing the microbiome. But there's a global reduction in the biodiversity. But for UC and Crohn's, they each have their own specific dysbiotic bacterial overgrowths that seem to be associated with IBD, which are listed there. And in both of them, they have an undergrowth of a very important firmicutes called Faecalibacterium prausnitzii, which turns out to be an incredibly potent anti-inflammatory probiotic. They lose this species. But there are many people who have dysbiosis, many people with genetic predisposition to IBD. They don't all get it. So there's a 3rd hit. And Fasano's work sort of has highlighted this. He believes it's permeability, altered permeability. See, there's an interface, right? Your gut bacteria are kept off your epithelium by a double layer of mucus and a layer of IgA. So they're there as a captive workforce to give us health benefits. On the other side of that gut barrier is your immune system and your genetics. Now, when that barrier becomes impaired, then bacteria are now coming in contact with your epithelial cells. This is not tolerated by our immune system. And if this is going on chronically, this permeability defect is where these two meet and where the problems occur. And it ticks all the boxes, this theory, in terms of the onset of autoimmune disease, right? So where are we at with inflammatory bowel disease in 2017? Well, we've got a reasonable way here. In ulcerative colitis, published this year in the Journal of Crohn's and Colitis, was a meta-analysis of 41 studies, some cohort, some randomized controlled trials. We've got 550 patients now that have been treated in research studies with FMT. And the overall clinical remission rate is 36% from summarising all those papers. Published this year in The Lancet was the Australian multi-centre study in UC. 42 patients with steroid refractory moderately severe ulcerative colitis. Now these guys were they really gave FMT a go here. This is a very intensive regime. It was 8 weeks of 1 FMT a day for 5 days a week. So this is the most intensive fecal transplant trial yet. Now they had an 8-week steroid-free clinical response rate, so a reduction in symptoms of 70%, but their clinical remission rate was 44%. This compares very favourably to the very expensive drugs we use like infliximab and Humira, which don't approach the success of FMT. Crohn's disease, we haven't got as much data. Here's an example of some of the cohort studies that were published a few years ago. 30 patients with refractory Crohn's. They were given 1 mid-gut fecal transplant, and they looked only at the 4-week remission rate, very high, 76%. We don't know what happened after that. This other study of 9 patients, again, 1 mid-gut fecal transplant, and they had 7 out of the 9 patients went into remission at 2 weeks, with 5 staying in remission at 3 months. Published this year was a meta-analysis of the 13 studies that have been done in Crohn's disease, a mix of cohort and randomised controlled trials. The overall clinical remission rate for Crohn's disease was 50%. This is very exciting, but it's not accepted like ulcerative colitis is. There are some professors of gastroenterology who are now saying we should be using fecal transplant for ulcerative colitis, but we haven't taken that step with Crohn's disease yet. We need more work. As you can see, it takes a lot of convincing, right? Over 500 patients minimum before we will pay any attention. Anyway, it's coming. So what's happening? Well, we know the best response seems to be the richness of your donor. The best biodiverse donors give you the best result. So what we know is that in IBD, you need to do multiple infusions, and you need to use multiple donors, and that means 3 or more in your sample. And obviously it correlates with, the patients that failed only had 1.5 phylotypes engrafted. The patients that responded, 5 phylotypes engrafted. So it really does work if you can accept the new microbiome. Irritable bowel syndrome. This is crying out for some microbiome modulation. Don't you agree? I mean, we know that at least 1/3 of cases of irritable bowel syndrome are post-infective. Some bug has messed up your microbiome and permanently changed it, and you're now stuck in this no man's land of irritable bowel syndrome. We also know that another 20% of those are post-antibiotic. So it's telling us that there's a microbial imbalance, there's dysbiosis driving irritable bowel syndrome. But there's very little data on it. No one's interested in irritable bowel syndrome. And this is not a drug, so there's no drug company money behind this. So we've got 2 studies only. One was in constipation predominant IBS. Now, we know there's dysbiosis in both IBS constipation predominant, IBS diarrhea predominant. That's well described. Okay, we can accept that. There are overgrowths of dysbiotics and there are undergrowths of probiotics. We can't argue with that. But 2 studies only of fecal transplant years ago. One was in 55 patients with constipation predominant IBS. 42% of them had a symptomatic improvement. 16% of those was a partial improvement, and 36% was a cure, total result disappearance of their IBS. A further study of 45 patients with a mixture of IBS patients, diarrhea and constipation. They had a course of fecal transplant, a very high response rate, 88% normalised their stools with disappearance of bloating and pain. And this effect persisted in 60% of patients between 9 and 19 months follow-up. Okay, so there's a lot of food for thought there. If you can effectively change the microbiome in irritable bowel, we might be able to cure irritable bowel, but it needs more work. Now, FMT regularly creates headlines, right? And here's another headline that was published in the Huffington Post last month. Could a gut bacteria transplant one day replace drastic weight loss surgery? And so everyone goes crazy when they see these headlines. Now, what they did was these authors published this study. They looked at obese patients, lap band and gastric bypass surgery, and they looked at their microbiome. They were thinking, well, God, people lose a hell of a lot of weight after a gastric bypass surgery, like a shit ton of weight, compared to anything else we do. But is it all just malabsorption? Somebody didn't believe it. So what they did, they looked at the microbiome. So we know that the microbiome of an obese patient is dysbiotic and it has very low biodiversity. If you do a fecal transplant from an obese patient into a 9-week-old germ-free rat, it gets obese. There is an obesogenic microbiome and it can be transplanted. If you do a fecal transplant from a patient who's had a human who's had a lap band, they get fat. But they're 26% less fat than the obese rats. So it's not fabulous. And the microbiome of the humans that had lap band surgery wasn't particularly good. But when you take the microbiome from someone who's had gastric bypass and you look at it, it's massively biodiverse. It's more biodiverse and bursting with probiotics, more so than a normal weight human. And you give a fecal transplant from a gastric bypass patient to a rat, it stays lean as 46% less fat gain than if it gets a transplant from an obese patient. So clearly with gastric bypass, there is an effect from malabsorption, but there is an ongoing effect from a lean microbiome that you've created. So everyone's wanting to look at those microbiomes. What is it that we can encapsulate and sell and make a fortune from? Anyway, so how do us humans, we control our own body weight? Well, our gut and our adipose tissue signals our brain, the hypothalamus pituitary with the arcuate nucleus, which is a little bit outside your blood-brain barrier. So it can signal the brain through these hormones, leptin, ghrelin, and insulin, to say you need more food, stimulate hunger, slow down your metabolic rate so you can gain weight, or the opposite. Now, it turns out your gut microbiome, your gut bacteria, they don't get fed unless you feed them. So unless you eat, they starve. So these little bacteria have developed ingenious little ways of making us eat. So what they do is when they want more food, they will secrete these signal peptides that induce the epithelial cells, specialized gut epithelial cells, to make another set of neuropeptides. PYY is one of them, GLP is one of them, and that these will enter the bloodstream and affect your pituitary. So you'll either be hungry or you'll be satiety, you'll be full. So they're telling you whether you're hungry or not, and you're telling yourself whether you're hungry or not. Now, certain probiotic bacteria, we know what they eat. They eat fibre, non-digestible starches, resistant starches. But what do dysbiotic bacteria eat? Sugar! Right. So they postulate that your sugar cravings in obese patients are not you craving sugar, but your gut microbiome craving it. Fascinating. So a team from Washington took 4 sets of identical twins, right? They had to find one that was obese and one that was lean. And what they did was they then did a fecal transplant from each of the twins into 9-week-old germ-free mice. Now, what happens on 2 weeks after getting a transplant, if you get the obese microbiome, the mice became obese. And they gained this blue column here, they gained a significant amount of body fat. Whereas if they got the fecal transplant from the lean twin, stayed normal body weight. Now, if you house these mice together, these transplanted mice, the fat ones and the thin ones, they are coprophagic like every other animal except humans. And the transfer of the microbiome only goes in one direction. The lean microbiome then took over the obese microbiome and the fat mice lost weight. It didn't go the other way. And that's because they were fed a good, healthy, balanced diet of rat chow, not McDonald's, right? Not much in humans yet. One study. This is looking at American men, only 18, who are obese, metabolic syndrome, and they looked at insulin resistance in these men. So they gave them a fecal transplant from a lean donor, meaning a BMI less than 23, and they found at 6 weeks, in the middle column there, after the FMT, their insulin sensitivity returned to normal. Now this happened despite no weight loss. So they found there was higher levels of butyrate-producing bacteria in the microbiome after the fecal transplant, which we know affects gluconeogenesis in the liver. So it was affecting the liver and improving their insulin sensitivity even without weight loss. But this can go the other way. This is another headline. This study shocked everybody. 2015 it was published, but in America, a woman with chronic recurrent C. diff infection persuaded her physician to give her a fecal transplant, which is not unreasonable. But she insisted on using her overweight daughter as the donor. I'm not having the FMT from anyone else. Unfortunately, pre-FMT she weighed 61 kilos, normal BMI, and at 16 months later she put on 16 kilos. She was now 77 kilos and with a BMI in the obese range. So they panicked, put her on a diet exercise program. At 3 years after the transplant, she put on another 3 kilos and weighed 80 kilos, an obese microbiome, BMI. So this, I mean, everyone goes, what the hell? You know, FMT's dangerous. You can't give it to anyone, you'll kill them. But this physician did not follow the Amsterdam protocol, which means you cannot use a donor who has a BMI over 26. So if you don't follow the guidelines, you know, if I give you too much iron, I'll kill you, right? Follow your guidelines. Then we're getting beyond the gut, obviously. Chronic fatigue syndrome, well, just about everyone with chronic fatigue has IBS. They've all got shocking guts. They've also got leaky gut, and you can measure levels of lipopolysaccharide in their blood that's getting across. And the dysbiosis is well described in their guts of patients with chronic fatigue syndrome, with overgrowths of certain dysbiotics and undergrowths of probiotics. So we've got one study. 60 patients with chronic fatigue were given a course of fecal transplant, and 70% of them had a significant improvement in their fatigue. And that lasted, at 18 months, 58 of them still had relief. At 3 years, despite no other manipulation of their microbiome, 35% had still had no chronic fatigue. So this is very interesting, and it's a whole lecture talking about that, but we'll just move on. Autism is associated with dysbiosis, particularly in overgrowth of Clostridial species, and some papers have shown that if you treat autistic kids with vancomycin and kill the Clostridium, they have an improvement in their behaviour. Tom Brody's reported 5 cases where he's used fecal transplant therapy in autism and shown a significant behavioural change, but that's all we've got. I love it. MS is fascinating, right? So the dysbiosis in MS is an overgrowth of an archaea species called Methanobrevibacterium smithii. Why is this bacteria in MS? Well, it turns out this bacteria's got cell wall antigens that are identical to myelin basic protein. So if this is getting through your gut into your bloodstream and you're making antibodies against this, they will cross-react through molecular mimicry against myelin. So this is kind of thought to be very one of the underlying causes, perhaps, of MS. So we've got little data again. We got a case report of 3 cases with severe MS. These patients had indwelling catheters. These patients were partially wheelchair-bound but had to walk with assistance. And they all have shocking constipation, the MS patients. So they had a course of fecal transplant. Now, why this got reported was because it didn't just cure their constipation and regulate their bowel habit, but all 3 of them had such an improvement in their bladder function, they were able to remove their indwelling catheters, and they were able to walk with aids and throw away their wheelchairs. So a significant improvement. And there's been nothing on this since 2011. No one's interested. I had one lady come to my clinic and she'd read this and she said, I've been told by my doctors I'm not responding to any treatment and that I've bought a wheelchair. My neurologist said there's nothing more we can do. You're going to be wheelchair bound. Anyway, she was shocking constipation, so I did agree to do the fecal transplant course on her. Thankfully, it normalised her bowel function, but she wasn't interested in talking about that anymore. She came into my room the first time I saw her. She was pushed in a wheelchair. And this woman came in to see me, 3 or 4 months later, makeup, lipstick, shoes, walking. I said, oh, I've got another patient with the same name as you. She said, no, that's me. Fuck, this blew me away. I said, no, she was in a wheelchair. She said, I was in a wheelchair. She said, before you did the transplant, I'd been assessed by my physio, my neurologist. I could walk a maximum of 60 metres with a 4-pronged wheelie frame at my absolute best before I either fall over or collapse. And I said, God, what are you doing now? I said, where's the frame? She said, I don't need it. I can now walk 600 metres without any aids at all. Now, her neurologist said that was a spontaneous remission. I kid you not. That's because we don't have enough data, right? We need to do these studies. Parkinson's disease. This is weird, this disease. So this is described as an alpha-synucleinopathy, right? So alpha-synuclein is a nasty little protein that deposits all throughout the brain in Parkinson's disease, causing the neurology. But guess what? Parkinson's disease patients all have chronic constipation, IBS, years before they get neurological symptoms. And biopsies of their gut show that there is alpha-synuclein all throughout the enteric nervous system in the gut, years before the onset of neurology. So one of the theories is that this acts like a prion, this alpha-synuclein, and spreads up the vagus nerve, and that's how it gets to the brain. So people are looking hard at dysbiosis. What bacteria is producing this protein? We don't know yet. There's some evidence to show a loss of prevotella, and if you've got gait and balance abnormalities, that phenotype, you have an overgrowth of enterobacteria. If you've got more rigidity and cogwheeling and tremor, you get an overgrowth of Ralstonia. This needs a lot more work. We've got one case report of fixing constipation with fecal transplant in Parkinson's, but a significant improvement in gait and mobility and balance. I've had one Parkinson patient come to see me, same deal, constipated up to the hilt. We did the fecal transplants, cured his constipation, his bowels regulated, and he had a significant improvement in his postural stability and his walking, but he didn't have much improvement in his cogwheeling or tremor. So we may be able to moderate certain phenotypes in Parkinson's if you change their microbiome. We need more data. And finally, ITP. So one case report was published in a patient with ulcerative colitis who wanted to have fecal transplant for the colitis. Now, colitis induced remission, but it also cured the ITP. So the platelet count went from 97 to 195 off all immunosuppression and maintained it. So there's no more work in this yet. There's currently an application before ethics in China looking at probiotics in ITP, but nothing looking at transplant. There's also a big study in Parkinson's in America underway, funded by the Michael J. Fox Foundation. So the data's getting there. What's the future? Well, we're looking at different ways you might deliver a fecal transplant. Fresh is still best. Frozen, we can add a cryoprotectant now, and you can freeze it at -70. It still retains its potency. They're looking at freeze-dried forms of it. They're looking at capsules, poo capsules, which are currently being tested in this country for C. diff, and you can get them in America. We will have better ideas of how to match a donor with a recipient once we know more about the microbiome and which bacteria we're trying to get. So we might be able to have Faecalibacterium prausnitzii rich donors, like super donors. And certainly for obesity, any study we do in that, they should be ultra lean. People who can eat a lot but are ultra lean. So we'll see a bit of that, and hopefully we'll see trials in autoimmune disease, mood disorders, autism, etc. in the future. And we might see isolation of microRNAs or bacteriophages being used in clinical practice. So that's where we're at. Thank you.